I got mixed up.
Okay, welcome back, everyone. It's actually just so nice to see us at a meeting together, conversing. This was the goal, was to actually have a little bit more conversation, and it seems like we're on the right track. When I was first thinking about who would we bring together for this set of talks around the use of botanical gardens and arboreta to do research, I thought in terms of keynotes immediately of someone who I think could lay claim to having almost as many papers published as Michael that involved the Arnold Arboretum. And actually, if someone wants to actually find out, I will have a plaque made for the one who comes out on top. All right, I think there should be some competition. But
Peter Duff.
okay. Oh, yeah, Peter. Whew, yeah. So bottom line is, there are some wonderful people who have really made the Arnold Arboretum central to so much of what they've done. And Richard Primack, who is a professor at Boston University, has spent years in these collections. He is intimately knowledgeable. And Michael may have a small quadrant that's focused on viburnums and their relatives, and that's great. The opposite of that is that when you do things like Richard, the entire arboretum is under your study and care, and so I'm just so pleased for you to have you, Richard, giving today's keynote, and of course tomorrow's keynote will be Suzanne Renner. So without further ado, growing and vital role of botanical gardens in climate change research. I think no one has been more the face of that community and that effort than Richard.
Okay,
So,
thank you.
Thank you so much, Richard.
Thank you very much. It's a pleasure to be here. I'm speaking with a microphone, so hopefully you should be able to hear me better, and also on Zoom hear me better. So actually, when Michael made his claim about having more papers, I was wondering if he could really make that claim. But what I can say is that I and my students have probably published about more species at the Arnold Arboretum than anyone else, because we've done these very large surveys of flower or leafing out and et cetera, which I'll tell you about. I also want to thank Ned and the Arnold Arboretum and the organizers of the symposium for inviting me here today. It's a great honor for me. And I think that for me, this has a very special meaning in that this is also my 50th graduation year from Harvard University. So it has that meaning for me. So I've been doing this work for my career. And I just feel so glad that I chose this profession of being a botanist and a plant biologist rather than a medical doctor. Not to say people shouldn't be medical doctors, but I think for my choice, I'm very happy with it. I also want to just recognize the fact that when I was an undergraduate, I had a tremendous amount of help from the professors, and they guided me. And I want to particularly mention Professor Carol Wood, who was also your advisor. Michael, is that correct? Yes. So when I was an undergraduate and suddenly became interested in plants, he sat down with me for 1 or 2 hours every week to just talk about plants with me. And that was one of the reasons why I was able to learn so much about plants as an undergraduate. And I have maintained this kind of desire to help students for the last 50 years. So whenever students come in to talk to me about plants or someone wants to work on a project, I always will say yes. I will never say that I'm too busy or that I can't do it, because I always remember this just very dedicated professor who not only was a great botanist, but also was willing to help other people. And that's something that I think is really a model of what we should all try to do as educators and scientists. So I'm going to talk to you today about the growing and vital role of botanical gardens in climate change research. This is based to some extent on a paper that myself, Abe Miller-Rushing, Libby Elwood, Amanda Gallanet, and a variety of other colleagues did in terms of this paper. And it's also calling on research that really dozens of colleagues have done over the last 20 years on climate change research at botanical gardens. And this topic is particularly relevant because while botanical gardens provide great resources to the whole range of botanical types of investigations. It's really the study of climate change which has really invigorated research at botanical gardens. It's really a mission which they didn't have up until 20 years ago, and it's really just created a lot of energy for new research ideas. And I'm sure, as all of you know, climate change is about the fact that people, by burning fossil fuels and releasing carbon dioxide into the atmosphere and also cutting down and burning tropical forests, are causing all these changes in the environment, like warmer temperatures, changing rainfall patterns, and also increased levels of carbon dioxide. And this means that botanical gardens can be used to study these types of phenomenon of climate change. So why do this work at botanical gardens? Why are botanical gardens so special for studying climate change? Well, first of all, botanical gardens have the plants. So Michael, in his presentation, and Jessica in her presentation, they talked about the fact that plants have a range of species, but they actually have huge collections of thousands or even tens of thousands of plant species there. So if you're interested in comparative studies, they're the perfect place. So if you're interested in red maples, well, you can study red maples a lot of different places. But if you're interested in the range of maples or the range of oaks, botanical gardens are the place to go. The other reason to study at botanical gardens is that's where the botanists are. That's where there are big concentrations of botanists or people who know about plants. So if you want to study plant diversity, there are often people there who can help you to do it. And often the staff there also can do research. Also, botanical gardens often have amazing historical records, as Nen mentioned in his talk and Patrick mentioned, that botanical gardens have these records of where plants were collected and often a lot of information on phenology and other plant characteristics. Botanical gardens also have a tremendous numbers of visitors and volunteers. So botanical gardens are the unique place in society to teach people about the importance of plants and also to involve the people, involve volunteers in learning more research techniques in plants, and also engaging the public in taking actions to deal with the problem of climate change. A basic question of botanical gardens is, can we use botanical gardens to see the effects of climate change? And the answer to that is obviously yes. So if we look at weather records from places near botanical gardens, so this is from the Blue Hills Observatory, which is the longest running weather station in the United States, we can see that the temperature has changed. So the temperatures have increased by about 3 or 4 degrees centigrade over the last 170, 180 years in this region. And so as the temperatures have warmed, also as the rainfall patterns have changed, and as the carbon dioxide levels have increased, we could expect to see changes in the collection from historical observations. So despite the fact that botanical gardens are incredible places to do research, you would have to say that botanical gardens are still underutilized. In comparison with what botanical gardens have to offer, the resources that they have to offer, they're surprisingly understudied. There is a lot of research going in there, but considering the number of botanists, the number of people working on climate change, the research going on botanical gardens is still minimal. And it often doesn't take advantage of the unique species there. The studies that are there often don't take advantage of the incredible diversity of species on the grounds of the botanical gardens. And this is something that myself and my team, when we were working on this recent review article, we started wondering, like, why are they underutilized? And we'd have to say that one of the biggest reasons that people at botanical gardens are busy. So if you ask people at the Arnold Arboretum or the Morton Arboretum or the Garden in the Woods, what they're doing, they just will start telling you about all the things they're doing. And they're working hard, and they don't have often the chance to stop what they're doing and start interesting new projects on climate change or other areas. So the garden staff are busy, and the people who don't work at botanical gardens, they're often busy with their own projects. And a lot of people who are botanists or ecologists, plant ecologists, ecosystems ecologists, they haven't been trained to appreciate the incredible resources of botanical gardens, so they don't think about what they're doing. So they might be doing a study of red maples, but they don't realize that they could go to the Arnold Arboretum and study 60 different kinds of maples, all of which show slightly different characteristics from the red maple. So they're generally people don't appreciate the value of botanical gardens, and that's something that we're trying to make people more aware of. I mean, another issue is to what extent people are welcomed at botanical gardens. And I think Ned described the fact that the Arnold Arboretum is a very welcoming place, but that's not the case at all botanical gardens. At many botanical gardens, you go there and you say to people, oh, I want to collect some leaves or some branches or some wood, and they'll tell you maybe you can't do it. So some botanical gardens aren't as welcoming, and that tends to limit also research at botanical gardens. So in our review of the subject, we tried to identify 5 questions that we thought were important, that botanical gardens were uniquely important to investigate. First is, how does the variety of species vary in their response to climate change? If you're doing comparative studies, how do they vary? Second question is, how do individual plants that are growing on the botanical gardens, how have they changed in their response over time, historical time, to the changing climate? The third question is, how will plants respond to the future? If you take climate change scenarios and you project them onto the plants at the gardens, how will they change in the future? A fourth question is, how will we be able to protect plants in the future from climate change? And then the fifth question is, how can we effectively communicate these ideas to the public and engage them with the issue of climate change? And these are all things that botanical gardens are uniquely suited to address. So I just want to start off by talking about some of the unique features of botanical gardens. One, a very important one, is to what extent is there historical information which can be used to investigate climate change? And it turns out that botanical gardens, once you start looking at botanical gardens, they often have a wealth of historical information which can be paired with modern observations to detect the fingerprint of climate change. So often you have to really search carefully, you have to talk to a lot of people, you have to look in the library or the herbarium, you have to see what's around, but often there's a lot of stuff. And so, for example, from the Arnold Arboretum, they have a herbarium collection of, I think, more than 100,000 specimens, something like that, around 100,000 specimens. Around 100,000 specimens collected on the grounds of the botanical garden. And it turns out that a very large percentage of those herbarium specimens are collected when the plants are in full flower. They have dates on them, and they have the number of the plant which they're collected from. So, for example, there is a specimen of Rhododendron vasei, probably one of the most beautiful plants on the grounds of the Arnold Arboretum, which probably it's a little too early for it flowering right now. But so we can take these herbarium specimens collected in the past. So this specimen was collected on May 19th, 1938 from exactly this same plant. So, and this photograph was taken on May 3rd, 2010. So by matching hundreds of herbarium specimens against hundreds of contemporary records of when plants are flowering, you can document that plants are now flowering about 2 weeks earlier at the Arnold Arboretum than in the past. So this is something that you can do. Another example is botanical gardens often have enormous photograph collections, and they often have dated photographs, and sometimes, if you're very lucky, they're actually from individual plants, but mostly not. Mostly photographs are taken of particular plant species, but by matching collections of photographs with modern observations, you can again see the effects of climate change. And at the Arnold Arboretum, for example, there are vast collections, but interestingly enough, they're in different places. So you have to kind of do a lot of detective work. You have to go find the collections in the library. You have to find the collections in different special kind of offices in the botanical garden. And a lot of the collections at the Arnold Arboretum are held by individuals, so they're their personal collections. So we have to find those people and look at their collections. And that by doing this, we were able to detect that just using photographs, you can actually see that plants are flowering earlier now at the Arnold Arboretum than in the past. And I should mention that this work was done, I mean, Abe Miller-Rushing was the leader in this, that Peter Del Tredici, who's back there, was also very involved in doing this study. And we got help from many people on the staff for doing this study and the earlier study with herbarium specimens. I also want to mention that these studies of using photographs and herbarium specimens to detect the effects of climate change were done in a botanical garden, actually the Arnold Arboretum. And these are now widely used techniques being used around the world, but also for investigating the effects of climate change on wild plants. So using herbarium specimens to look at climate change is a very widespread technique, but it first started here at the Arnold Arboretum. And this is an interesting photograph because this figure right here, which is from the Miller-Rushing 2006 article about using photographs to detect climate change, it shows one thing, which it shows that plants, by matching, each one of these symbols here represents a dated photograph. And by matching the dated photograph with when the same plant was flowering in 2003, you can see that plants are flowering earlier over time, as shown by the solid line. I put up here the dotted line of the same data, but using herbarium specimens instead of photographs. And the point of this is to show that herbarium specimens and photographs give exactly the same kind of data. They show the same relationship because they're the same kind of data, that herbarium specimens and photographs are both representations of when plants are in peak flower. So this was kind of a breakthrough to realize that these different kinds of data sets could actually be combined in climate change research. The other thing that I want to point out here, this is really weird data. So for those of you who've had classes in statistics, know that this is unusual because it's not continuous data. It's just the data tends to be concentrated in a few different dates. So often phenological data and data using herbarium specimens and photographs has a lot of unusual statistical properties that you have to consider when you're doing this kind of research. So just as another example of the kind of data which exists out there when you go looking for it is that at the Royal Botanical Gardens at Edinburgh, they have this just unbelievable data sets of rhododendrons. At this botanical garden, they've always been interested in rhododendrons. So they have all these herbarium specimens that were collected in the Himalayas, and they brought back all these species to the botanical garden in Edinburgh. People have been gathering phenological data on flowering times since the kind of late 1800s and up into the present time. They have all these herbarium specimens collections, and then also they have this very active group of volunteers, of citizen scientists, that likes to go out and monitor the flowering time of rhododendrons. So it's this unbelievable data set that they have, which is now being repurposed for climate change research. So we talked about individual botanical gardens, but botanical gardens, there's a lot of botanical gardens in the world. And so it's possible, if you're interested in climate change, to investigate climate change at many different botanical gardens and to create networks. But if you look at the position, the locations of botanical gardens shown here as red dots here, you can see that they're not evenly distributed around the world. They're really concentrated in Western Europe and North America, and in particular in Eastern North America. And if you look at large areas of South America, Africa, Western Australia, and kind of northeastern Asia, there really aren't botanical gardens in these areas. So botanical gardens tend to be very unevenly distributed around the landscape. And this lower figure here shows where there are the number of species held in botanical gardens. And again, you can see that kind of Europe and Eastern North America, and to some extent California, the West Coast, that's where there are great concentrations of species. And the botanical gardens which exist outside of this region tend to be smaller in size. So a very uneven distribution of gardens, which limits our ability to do global climate change research. So the real secret to doing climate change research at botanical gardens is to link them together so you have greater numbers of species and a greater number of locations. This is actually a point that Patrick made so effectively in his talk just earlier. And a lot of these botanical gardens are now being linked together to make phenological observations. Jessica already defined phenology, but it's the timing of events. Such networks exist already and are being expanded often for specialized types of global climate change studies. One of the most well-known is the International Phenology Gardens Network, where people planted special phenology gardens at botanical gardens. We established a network in 2011. It's actually interesting that we never gave our network a name. So we just organized a network of people to talk about, to investigate the phenology of woody plants but we never actually gave it a name, which is, so it seems incredible to think, like, why didn't we give it a name? We just called it, like, people monitoring woody plants. So we had the Woody Plant Network, which consisted of 7 botanical gardens. And now there's a recent botanical garden, which is very exciting, called a recent botanical garden network called Phenops, which has just got going about 3 years ago. The position of these gardens is here. And this is a special garden just to look specifically at perennial herbaceous plants that we could call wildflowers. But it's a network specifically focusing on neglected wildflower species. So a real shift in terms of people investigating climate change and phenology over the last few years is not just focusing on flowering time. In the past, people focused primarily on the flowering times of plants for phenology and climate change research, but there's been this shift now toward people looking at leafing out times, as Jessica described, also people looking at fruiting times of plants, and also very, very recently looking at leaf senescence times, which has really been a neglected characteristic of plants. So what I'd like to describe to you today is just some of the main findings of this network on woody plants that we established about 10 years ago. And in this, we did 3 interrelated studies of leafing out, leaf senescence, and fruiting times. So we started off, the first study we did was of leaf out times at 7 botanical gardens. And if I actually tell you how this network was organized, it's really unbelievable. I sent an email to Peter Del Tredici like in February, I think of 2011. I said, what are some really great arboretums around the country? And so he just gave me a list of about 8 or 9 botanical gardens that were really great. And I sent emails to, I think, about 7 of them, besides the Arnold Arboretum. And within a couple of days, most of them replied, including Suzanne Renner from the Munich Botanical Garden. They wrote back and they said, sure. There was one botanical garden that said they were too busy, but most of them just said, great. But first of all, nobody ever asked about money. No one said, like, how is this being paid for? No one said, like, who's going to be the author of these things? No one really just had any of these logistical questions. They just said, great subject. We want to get involved. We want to do it. And everyone came on board. We had no money, no budget, and we also didn't give it a name. We just said, everyone who's just going to go out and monitor woody plants. So we started off in 2011, and we just agreed that we would record for different species when they made their new leaves. And we defined it very simply. So you record a species when you could first see the shape of new leaves on a plant. And we just got amazing results. I mean, it was so exciting that Jessica referenced Lekowitz's paper from, I think, 1984. So Lekowitz was the largest study of leafing out times of woody plants prior to that. And he had records of about 80 or so plants. And we recorded over 1,500 plant species, over 1,500, the leafing out time of over 1,500 species. And for many of those species, they were recorded in 5 or 6 or more of the botanical gardens that we were studying. And we found that there were incredible differences when you actually went out, for example, at the Arnold Arboretum. So if you go out on the grounds of the Arnold Arboretum, in a normal year, some plants start leafing out in early April, and then they just continue on through the end of April, May, and then plants are even leafing out, especially, for example, pines are leafing out through the middle of June. So a tremendous variation in leafing out time, which had not been appreciated. Also, that there are very strong differences between genera and families, a very strong phylogenetic component. So, for example, the Rosaceae tend to be almost entirely very, very early flowering or very early leafing out plants, whereas, for example, the Fagaceae or the Leguminosae tend to leaf out really late, which hadn't been appreciated. There's some really larger taxonomic differences. So, for example, gymnosperms leaf out later than comparable angiosperms. And then there's also some habitat effects. So, for example, deciduous species leaf out before evergreen species within the same genus. So, for example, in rhododendrons, shrubs leaf out before trees within the same genus or the same family. And then there's a difference, which I'm going to foreshadow Suzanne's talk tomorrow. So I think she's going to tell you that using this large database and additional data from Munich, that there were differences between biogeographical regions. But then you can also apply modern phylogenetic analysis to this. So using modern techniques, you can't just say, well, there's average differences between families in general. You can actually take this data and put it onto a phylogenetic pattern. Unfortunately, Chuck Davis is not here, but Chuck Davis and his student at the time, Charlie Willis, collaborated with us in this paper led by Zoe Panchin. And we took all of our data and we put it onto this modern phylogenetic analysis. And what you can see here is that the dotted line here represents the average leafing out time of the plants. The gray dots are the plants that leaf out early, and the black dots represent the plants that leaf out late. And you can see that there's a lot of variation in leafing out time, but there are these clusters. So that one, for example, shown right here, this is the rose family, and they tend to leaf out very early. 3 here is the oaks, and they tend to leaf out very late as a group. And then if we come across here to the gymnosperms here, that you can see that the gymnosperms tend to leaf out really late. But what's interesting is that the triangles are evergreen species. So these are the evergreen gymnosperms, and they leaf out really late, like pines and spruces and firs. But that the few gymnosperms that leaf out early are the deciduous ones, like larches and ginkgos. These characteristics of leafing out time are linked to wood anatomy, as Jessica has already mentioned. So this is a very large sample size, again showing that, that species with diffuse, porous stem anatomy universally tend to leaf out early, and species that have smaller diameter vessel elements, again, tend to leaf out early. So confirming what she just described. We've also done studies of leaf senescence on the grounds of the Arboretum. And at these other botanical gardens, we had this network. And leaf senescence is very important because leaf senescence represents the end of the growing season, the end of the time when plants are absorbing carbon dioxide, the end of the time when plants are producing carbohydrates for growth. It also represents a very important linkage to insect and bird populations and to microclimate and various other ecosystem processes like evapotranspiration. But curiously, the time in which plants undergo leaf senescence has been neglected. In particular, the variation among species in terms of when they lose their leaves has been seriously neglected as a subject. And so beginning in 2012, our Woody Plant Network began to look at the leaf senescence time of 1,360 species on the grounds of 6 botanical gardens. And these are pictures from the Arnold Arboretum here. And the first big question that we had to address is, what do you measure? So if you're looking at over 1,000 species, what is the measure of leaf senescence? And what we decided is that leaf senescence was when 50% of the leaves had done something, whatever that something was, whether it was falling off the tree, whether it was changing color, or some combinations of them. We also decided we weren't going to agonize what 50% was. So if you looked at the tree and we said, it's about 50% or more than 50%, we would call it that date. So for example, with Paulownia, the leaves mostly fall when they're green, or for example, for this mulberry species, the leaves are all turning yellow. So we use this 50% category. And one of the key results of this was that there's a huge amount of variation in when different species are senescing at botanical gardens. So among the 1,000 or so species which we followed at the Arnold Arboretum, that species varied from when they underwent senescence from September all the way through December and even January. So we have this Chimonanthes here, which is actually undergoing senescence after the first snowfall has occurred. The pattern of leaf senescence is so different from the pattern of leafing out. So what we see here is that there is a correlation from year to year at the same botanical garden. So the sequence of senescence is correlated from year to year with the same botanical garden, but it doesn't correspond to different botanical gardens. So what's happening here at the Arnold Arboretum is different from what's happening in Berlin. And similarly, there's a phylogenetic pattern in some botanical gardens, but not others, and often a different phylogenetic pattern. And the reason for this is that botanical gardens do something really interesting, which is that they plant all the related members of a species, often in the same place, like here at the Arnold Arboretum. And what this means is that often closely related species are in a similar microsite. So sometimes they're planting all the plants of a species in a wetter place, or a drier place, or a sunnier place, or a shadier place. So because they share similar microsite conditions, it often affects how they undergo the senescence process. So, and that's why we don't see any correlation between gardens, because the plants, the same species, are often in different microsites in different botanical gardens. And so what this means is that the patterns of leaf senescence are much more complicated than the process of leafing out. So the process of leafing out is largely determined by phylogenetic considerations and by habitat considerations. And leaf senescence characteristics are really determined much more by microsite conditions and by the way in which a botanical garden is managed, and by the year-to-year variation in rainfall patterns and temperature. And so as a result, we don't have these strong characteristics of species the way we have in leafing out times. So I would like to mention a third study of fruiting times at botanical gardens. This was done at 5 botanical gardens with over 400 species of plants. So done at the, just to kind of talk about the individual botanical gardens, so the Arnold Arboretum, the Botanical Garden of Berlin, the U.S. National Arboretum in Washington, the Beijing Botanical Garden, the Morton Arboretum near Chicago. And this was primarily led by Amanda Gallinet, a graduate student at Boston University. And the first thing which we had to decide on is what we were going to measure for fruiting phenology. And there was really no standard protocol for looking at fruiting time. And so what we had proposed is that people would monitor when they saw the first ripe fruit of a plant species, when the plant started to have at least 50% of its fruit ripe, when less than 50% of the ripe fruit were still present on the plant, and when the last ripe fruit was present on the plant. So we measured all these 4 characteristics. So we not only wanted to look at fruiting phenology, but to try to determine what was the best characteristic to use. So what we found is that there was a very strong ordering of species between successive years at Botanical Garden, and the sequence of fruiting of these 400 species was virtually the same at all of the botanical gardens we investigated. And also we found that the first fruiting time predicts the peak onset. So the first fruiting time was very highly correlated with the beginning of peak fruiting and the end of fruiting. So that means if you're doing this kind of research, you can just measure one statistic, which is when you see the first ripe fruit, and you're finished. You've got most of the information that you want. We also found that there was a very strong phylogenetic effect in fruiting times. So for example, we had groups like cherries and shad bushes and Amelanchier species tended to fruit very early, and species like Euonymus and privets were fruiting very late, very strong phylogenetic effect. There were also strong phylogenetic effects of duration. So for example, shad bushes tend to have a very short fruiting window, whereas things like privets tended to have a very long fruiting period. And this type of information on fruiting phenology and fruiting duration then informed subsequent work that Amanda Gallinet did on the feeding preferences of native birds in this area. So she did work at the Manomet Bird Observatory, and again seeing what birds were eating and when fruits were available. So if we take these 3 studies and we sort of think about what are the main messages of these 3 studies, one of the things that they reveal is that there's a tremendous amount of natural history and ecological and botanical information at these gardens. So no one had ever tried to do these kinds of large-scale surveys before, and we found that there was just this huge amount of really undiscovered information which was available once you started to look in these types of studies for information which was relevant to ecology and climate change research. We also found a lot of new methods. There were a lot of methods that could be used in our studies, which now people are applying at other botanical gardens and other types of studies on wild plants. There were a lot of new research questions that were generated. So questions, for example, that could be then addressed with further studies in physiology or anatomy or related studies on insects and birds. The other important point is that whenever we published these studies, we made this data available online, and especially with these large-scale studies with many botanical gardens, if you make the data available online, people will use them in further studies and will link them up to other types of botanical studies. So another very important follow-on from our woody plant network is that when we completed this, we realized that we had focused all this attention on woody plants, and that herbaceous plants had been comparatively neglected in botanical garden phenology studies. There's a wealth of information from botanical gardens on when herbaceous perennial plants, which we sometimes call wildflowers, so a tremendous amount of information about when they flower, but surprisingly little information about when plants leaf out in the spring, when they mature their fruits, and when the leaves undergo senescence in the autumn. So 3 years ago, we decided to form a new network. In this case, we gave it a name. We called it Phenops. So the Phenops network is a network specifically focused on the phenology and climate change research associated with perennial wildflower species. It's primarily based in Germany. The leader of the group is Christina Romerman from Jena. But there's another series of gardens and universities in this area around Berlin and Leipzig who are involved in this. And it now includes an international network which is expanding. And again, people making standard observations on the phenology of perennial wildflowers. So these botanical gardens are really focused on monitoring phenology in a very standard way on about 212 wildflower species, but are expanding it to include a larger network. So they include looking at the flowering time, leafing out time, leaf senescence time, and fruiting time of these perennial wildflower species, but also a very strong linkage to life habitats like the plant size and leaf area, and also in functional characters. So a very strong emphasis on things like leaf thickness, nitrogen content, carbon content, etc. A lot of effort in this project initially went into how do you record the first growth of perennial plants. And this is a challenge that you don't face when you work on woody plants, like at the Arnold Arboretum. So with the Arnold Arboretum, when we were interested in leafing out, we just go out and there's this tree with a tag on it, and you just see when the leaves open. But when you're working on perennial wildflowers, you're out there looking on the ground, and then you see something poking up from the ground. But is it the plant? What plant is it? How do you identify that plant? And if there's a lot of plants in a garden, how do you know which species there was? You know that there was a certain milkweed in that place last year, but is that what you're looking at coming up now, or is that milkweed now over there? So a lot of challenges of figuring out what you're working with perennial wildflower plants. And also the wildflower plants, it isn't just that their leaves expand, they sometimes come up with the stalk and then the leaves expand. So it's a challenge to figure out how you measure first growth in the case of perennial wildflower species. So this is a figure from it's a figure from a paper of ours which is currently in review. And in this figure, it highlights some of the most important factors which determine phenology for different stages of plant growth. And the one which I want to particularly point out to you is the characteristics of individual gardens, the variation in gardens. And so if you look at initial plant growth and the timing of leaf unfolding, each one of these factors here represents their relative importance in determining this phenological characteristic. And when you look up there and you look at initial growth, you can see it's gardens, the characteristic of gardens, which are very important in determining growth. And so even though these all represent botanical gardens in the Leipzig, Berlin area, that it's actually differences between botanical gardens in the amount of rainfall they get, and also in terms of how they cultivate the plants, whether it's open or sunny or shady, and whether they water or weed or not, which determines when you have leaf out occurring in the spring. That's the major determinant of variation in leafing out among species. But on the other hand, if we look at flowering time, so for example, the onset of flowering, it's really plant height which is the most important. Plant height is the dominant factor. So tall plants tend to start flowering tall plants tend to start flowering later in the year, and short plants tend to start flowering earlier. If we look at the onset of fruiting, again, it's plant height. If we look at the duration of fruiting, it's actually leaf area. So plants that have very big leaves tend to have a very short fruiting period, and plants that have very small leaves tend to have a very long fruiting time. If we look at the timing of leaf senescence, it's the plant height which is the most important. So plants that are very tall, their leaves tend to be lasting much longer in the autumn than plants that are very short. So this is a series of different kinds of characters which are correlated with these phenological stage. A very important part of the result here is that a lot of these very fine characteristics were measured, like things like specific leaf area, leaf dry mass content, nitrogen content, carbon content, but these were much less important than these characteristics which are relatively easy to measure, like plant height and the size of the leaves. So this tells us that a lot of phenological research can be done in a relatively straightforward without a lot of technical methods. I also want to mention another great opportunity which is opening up at botanical gardens, which is the use of herbarium specimens. So herbarium specimens, botanical gardens often have large numbers of herbarium specimens, but increasingly there are millions of herbarium specimens which are now being digitized and available for research, and they can often be used in a very creative way at botanical gardens in climate change research. And for example, herbarium specimens often, besides looking at them for flowering time, which people have done for a long time, they can also be used for looking at leafing out time. So you can look at this maple specimen, and it has young, expanding leaves that are translucent, indicating that it's a young leaves on it. And so by looking at dated herbarium specimens, you can get leafing out times from botanical gardens and also from the wild. And so this, actually, I just threw this in. This is kind of an unpublished study of mine from 15 years ago. And I like this figure, and I sort of just never got around to publishing it. But one day I went into the Arnold Arboretum herbarium, and I just looked at specimens that had young leaves on them, and I wrote down the dates of them, and then found out the temperature of the spring for those particular years. And you can just, if you just look at the X's right here, which represents Heliza treptraptera. So the X's represent the flowering dates on the y-axis and the temperature in the spring for a particular year. And you can see they're all falling on these just nice lines here. That when it's a very cold spring, they start leafing out late, and when it's a very warm spring, they start leafing out very early. So this is just kind of a simple thing you can do with herbarium specimens just very easily at the Arnold Arboretum. But there's a lot more things you can do, and I'd like to describe another project using herbarium specimens to look at anatomy and physiology. This is work that Abe Miller-Rushing took the lead on. We have Pam Templer back there who's going to be talking tomorrow. Raise your hand, Pam. So it's Pam Templer who's going to be one of the speakers tomorrow, and Pam helped us on the physiological aspects of this work. So we were interested to know whether individual plants at the Arnold Arboretum, individual trees growing at the Arboretum, had changed over time in terms of their physiology in response to changing CO2 levels and changing temperatures. And our hypothesis is that as the CO2 levels in the atmosphere had increased, that we would expect that plants would be needed to produce fewer stomates because they were reacting to higher CO2 levels by having fewer stomates. They would still get the same amount of carbon dioxide. And that also plants would be less efficient in terms of water usage because they could get carbon dioxide much more rapidly inside the leaves. So what we did is we looked at herbarium specimens collected over the last 120 years for trees still growing on the grounds of the Arnold Arboretum. And we went out and we gathered herbarium specimens from the same trees. We did that for trees of maples, oaks, and hornbeams. We used the old nail polish technique where you just paint the leaves with nail polish. The undersides of the leaves, you paint them with nail polish, and then you peel off the nail polish and then look at it under a really good microscope. And we also took leaf fragments, and we looked at them for stable isotopes to determine the water use efficiency of those leaves. And these are just some of the results. So the main result was that the stomate density declined over time, and you can see that by looking at two images from the same tree. What tree it was, I don't have to look at the paper, but these are two images from the same tree, and you can see that the density of stomates has declined from 1931 to 1985, but correspondingly, the stomates have gotten bigger. So overall, the stomatal conductance has not changed, but there's been a change in this ratio of density and size. But the water use efficiency studies showed that there had been no change in water use efficiency over this period of time. So contrary to our hypothesis. Another great opportunity for botanical gardens that we and others have taken advantage of is using dormant twigs to look at physiological responses to climate change. And it turns out there's an extremely easy technique that we and others have started to use. Lizzie Walkovich, who was at the Arboretum for a long time, was also using this technique very actively. And this is a very simple technique where you collect dormant twigs in January, February, March, and April, and you bring them inside, and you see whether they leaf out or not, and how long they take to leaf. And this helps you to determine, their characteristics help you determine what the winter chilling requirements are of those species, the photoperiod requirements, and the spring forcing requirements, or the spring warming requirements. So these things that plants need to detect the spring and to detect the change in climate is, in fact, it's a very, very easy technique that you can do even without specialized growth chambers or greenhouses. Just bring plants in and just see how long they take to leaf out. And this is kind of just an interesting example right here. So these are beech twigs, and these beech twigs we just brought in sometime in the early spring, and we just put them on a counter, and they didn't do anything in the regular day length. And then these plants, we had supplemental lights over them, so they had extended day lengths, and they leafed out. So what this tells us is that beeches, besides a winter chilling requirement, also have a photoperiod requirement, and if you expose them to very long days like late spring, they'll start leafing out. But this is actually the exception. So actually, the beeches is the one species that has a very strong photoperiod requirement. So in addition to kind of looking at these requirements, we can also specifically investigate the effects of temperature on spring temperature on the leafing out time of plants by putting plants in a wide variety of different temperature conditions. And this is a really great technique for investigating the effects of climate change, but it's also a great study for children, for university students, for middle school students, to just teach them the effects of climate change. So this is a crabapple where we gathered twigs in March, and we put them in a range of temperatures going from 4 degrees centigrade to 25 degrees centigrade, and we just looked at the effects of those temperatures on leafing out time, and you can see that when you put them in 4 degrees centigrade, they're mostly not doing anything after a couple of weeks, but when you put them in 25 degrees or 21 degrees, they start leafing out and flowering very rapidly. So it's a very effective way to demonstrate the effects of climate change. So at this point, I'd like to sort of try to finish up and just try to describe a number of promising directions in climate change research that can be done at botanical gardens. One is that we just have to recognize that we have this unequal distribution of botanical gardens. We need to try to encourage there to be more botanical gardens or more research in tropical places. Or for those few botanical gardens in the tropics, we need to try to reach out and collaborate with them in climate change research. We need to continue monitoring on the ground, so existing monitoring studies need to be continued at all costs. We need to maintain monitoring schemes and set up new monitoring schemes. We also need to increasingly digitize those records that are available. So again, botanical gardens like the Arnold Arboretum or other big gardens, I mean, in addition to digitizing herbarium specimens, digitizing records, digitizing photographs, and making them available to a wider audience. We also need to take advantage of the increasing millions of herbarium specimens being digitized around the world. And this is an example of what can be possible. So I'm collaborating with a postdoc named Ben Lee, who's at the Carnegie Mellon Museum, or excuse me, at the Carnegie Museum. And we, with a group of collaborators in Germany and in China, are looking at digitized specimens of trees and wildflowers and determining when trees leaf out and when wildflowers are flowering. over large geographical areas to look at biogeographical patterns. And each one of these symbols here represents a herbarium specimen. So in these 3 large biogeographical areas, we've been able to find 5,000 specimens which have the right characteristics that we want. And then again, we're using these to see the effects of climate change already and predict what future climate change will be like in these areas. And then we need more integrated studies of climate change. So in addition to just measuring phenology, as Jessica has mentioned, we need to combine that with studies of physiology and anatomy. We need to include all stages, not just flowering, but increasingly focusing on leafing out and leaf senescence. And also, again, we need to have more emphasis on wildflowers because they've been comparatively neglected. And then one thing which I haven't mentioned, but Patrick certainly mentioned in his presentation, we could have a whole lecture about, is the value of botanical gardens in plant conservation. So botanical gardens are very important because they contain genetic variation of species. They also represent a place to study the ecology of rare species. If you want to protect them in the wild, you can study them first in botanical gardens. It's a place where we can also start thinking about managed translocation, where we can bring plants further north of their current range as a way of seeing whether of using them to track climate change. In effect, botanical gardens are doing that already because botanical gardens are growing species from further south, and we're seeing whether they can grow on the grounds of places like the Arnold Arboretum. And if they start growing better here, that gives us an indication that they're starting to adapt to the kind of warming climate that we have around here. Another really important opportunity is using botanical gardens to potentially looking at frost tolerance of plants. As Jessica mentioned, that when plants start leafing out and flowering earlier and then there's a late frost, plants are more susceptible to frost damage than in the past. So looking at frost tolerance is a very important opportunity for botanical gardens, but also looking at things like drought stress, heat stress. So when we have unusual heat waves in the summer, to what extent will a collection like the Arnold Arboretum collection be able to survive these especially warm conditions? So actually I have a picture here of the coast pepperbush right here, and it's just showing the coast pepperbush last summer when we had a period of very hot weather and its swamp habitat dried out. And then another opportunity is linking up the kind of research going on at botanical gardens to the kind of ecosystem research that is very exciting right now, the kind of research that maybe Pam Templer will be talking about tomorrow. And so people are very interested in all these ecosystem processes like carbon dioxide levels in the atmosphere and the ability of plants to grow, hydrology. And this is something where comparative studies at botanical gardens could link up with these ecosystem studies. Just as an example, the fact that native shrubs tend to leaf out later than non-native shrubs, and non-native shrubs tend to hold their leaves longer than native shrubs in this area. And what are the ecosystem's implications of that for things like carbon budgets and photosynthesis? A recent paper that we published are suggesting that trees are more responsive to climate change than perennial woodland wildflowers. And again, thinking about that in terms of its implications for ecosystem studies. And then another important future opportunity is increasing use of technology, of drones, fixed cameras, and other other monitoring technology in order to be able to monitor things like phenology. And then finally, a great opportunity which presently exists and which Ned referenced already is the role of botanical gardens in educating the public about plants, about incorporating citizens into volunteer projects to monitor things like phenology and other characteristics, and also to engage with the public in terms of climate change and dealing with the problems of climate change. I also want to mention the fact that there are these great opportunities in terms of platforms and systems for getting volunteers to contribute observations like the National Phenology Network or the iNaturalist Network. So botanical gardens can develop their own technology to incorporate volunteers, but there are these existing online networks, like, for example, iNaturalist, where people just can go around a botanical garden and take photographs, upload them to iNaturalist, and then it also becomes available to the botanical garden itself. Okay, so what does this all mean? So I hope I've convinced you that botanical gardens are great places to do research in general and great places to investigate climate change and to communicate with the public. I hope I've convinced you that historical records can be used historical records combined with modern observations can be a really great way to investigate climate change and also to predict what's happening in the future. And I hope I've convinced you that we can gain insights from botanical gardens which are very relevant to climate change. Thank you.